All right, I'd like to thank the organizers for the opportunity to present this talk on some of the work I've been doing on some new methodology for measuring nutrient uptake, distribution, and localization in apples, and I'll talk about how it might apply to perennial fruit crops as well. So the basic outline of my talk is I'm going to present some methods that I've been using to understand nutrient distribution, going from microscale or cellular scale on an individual fruit, to a whole fruit distribution, to whole tree distribution, and then working within the orchard. And we're going to have a panel discussion at the end of this session on remote sensing and nutrient diagnostics. And so I left out the whole orchard aspect because I think it fits well within the panel discussion. So I'll focus primarily on the microscale, the individual fruit, and the tree nutrient distribution. And I'm going to present some work we've been using isotopes and analog tracers to look at nutrient uptake and distribution in apples. And then relating both of those previous aspects to calcium-related disorders and fruit cellular structure and how all that relates to the development of disorders in apple. So the challenge with nutrient-related research, as you all know, is that, especially with perennial fruit crops, existing bulk nutrient pools are very large, and they're difficult to distinguish from newly acquired nutrients, which that pool is relatively small. So when you do an experiment looking at dose responses, or looking at uptake, or looking at a new strategy for managing nutrients, the observed changes between treatments are often very small. And to add on to that, there's confounding orchard factors that add variability to your results that exceed these small differences. So very often there's variability in our replicates among trees that add variability to the results we see. Factors like crop load or soil variability within an experiment, irrigation variability in irrigation delivery aspect, topography. So there's a whole lot of variability that is added to these experiments that very often will lead to noise exceeding the changes that we see, which is then difficult to run experiments that see meaningful results. So for some of the work I'm going to present today, I wanted to highlight these government user facilities that are available for researchers around the United States, around the world. I have been using in the United States the Advanced Light Source, which is at Berkeley near San Francisco in California. And also I've been using the Canadian Light Source, which is a synchrotron facility in Canada, in Saskatchewan, Canada. And both these user facilities follow a similar type of call for proposals that is similar across all of these different government synchrotron facilities, where every 6 months they put out a call for proposals, and anyone is welcome to put in an application to get access to Beamlines. And Beamlines are these specialized high-powered tools that can be used to interrogate things like cell structure or nutrient distribution or crystallography of proteins or protein interactions or spatial mapping of nutrients. There's a whole lot of things that could be done within these individual Beamlines. So you put in these proposals of what you're proposing to do, what your question is, what your hypothesis is, and then you put in these applications to get access to these Beamlines. And you do this every 6 months. And if you're successful, you'll get maybe 48 to 72 hours access every 6 months for 4 cycles. And in those 4 cycles, you do your experiments. So these are really small time frame type projects that help support other experiments or other ongoing experiments. And they really just provide access to the beamline and expertise to help run your experiment, do your experiment at those facilities and process the data. So carrying out the experiment beforehand and afterwards, and all of the costs associated with that are on the investigator. But these facilities just give access to these really specialized pieces of equipment that allow you to do that much more. So if you are curious about doing this type of work, I encourage you to reach out to some of these synchrotron facilities in the future around the world. So for our research locations, that I'm going to kind of present some examples of what we've done. We work at the Advanced Light Source at Lawrence Berkeley National Labs in just across the bay from San Francisco. You can see at the top left. We worked, so these are all the different beamlines that are present. We worked on 8.3.2, which is a tomography beamline, essentially giving high-powered micro CT or CT scans to individual fruit to look at subcellular structure. And then 10.3.2, which is the Xray fluorescence microprobe, which allows us to look at spatial variability and nutrient distribution. We've also done similar work with the tomography at the Canadian Light Source, and as well as the Pacific Northwest National Laboratory, where we had a collaboration to look at whole fruit distribution instead of subcellular distribution. We did that 2 years ago. So X-ray fluorescence is a way to look at nutrient distribution with almost down to the subcellular level. So the X-ray fluorescence works in the sense that X-rays are applied to a surface of a structure, and it excites the electrons for each individual molecule. And those molecules, when the electron basically kicks off an electron out of a K shell, and the energy at which those electrons are ejected from the shell, from the electron shell, is specific to a specific element. So calcium has a specific energy that those electrons are fluoresced at. So we measure fluorescence, or the emitted energy or fluoresced energy off of the surface that is exposed to an X-ray. And that tells us what elements are present. So we can see when we use the microprobe, which scans on a two-dimensional map on the fruit surface down to a subcellular level. I think this is 40 microns. We can see the calcium and potassium distribution on that fruit surface. So here we see a bitter pit lesion right in the middle. There's healthy tissue on the outside. This is a crack that formed when we took the sample and froze it with liquid nitrogen on Peltier, using a Peltier cooler. So that, cracking sometimes happens, but you can see that, so you see the crack, that's just airspace. And then in the middle, you see the bitter pit lesion, which is primarily calcium, because it's dead cells, and dead cells are leaky, and the potassium is actually leaked out and been captured. So you see this potassium, high amounts of potassium ring right around the outside of the bitter pit. And that's basically just capture of potassium that is leaked out of this inner dead lesion spot. Then you can see a mixture of potassium and calcium on the outside of the peel. We see that fruit with bitter pit compared to healthy fruit with bitter pit has a much more red or higher amounts of potassium on the outer healthy tissue than a fruit with bitter pit. And these little dots in the middle here, that is just residual calcium carbonate that we weren't able to wash off the surface. So visually, we washed the fruit with deionized water several times, but we still were not able to get all the calcium carbonate off the fruit. So there's still some remaining residue, and that forms these really bright blue specks in those images. But this allows us to really look at two-dimensional spatial distribution of calcium and potassium on the fruit surface, and compare that between a healthy fruit and a fruit with bitter pit. We can also, so that's on the fruit surface. We can also go from the outer surface to the inner part of the fruit. And we can also use a technology called microscenes. And that allows us to look at the speciation of calcium. So whether it's calcium citrate or calcium nitrate or calcium pectate, they all form different or fluoresce at different energies, slightly different energies, and they have different signatures. So based on that, we can look at the different types or speciation of calcium on a spatial distribution basis. So we can look at where there's more calcium citrate or more calcium nitrate or more calcium pectate. And this, again, a two-dimensional sample that's excised from the fruit and froze on a Peltier cooler. We can also use the imaging tools and sampling tools. So we have these 6 or 7 different spots on the fruit that were specifically interrogated, where we get the potassium to calcium ratio for the X-ray fluorescence. And you can see some areas have very high ratios and some have very low ratios. And we're working on how that might associate with formation of bitter pit or differences in cell structure. So I should explain the use of X-ray fluorescence in terms of examining subcellular distribution of potassium and calcium. There's also handheld X-ray fluorometers that are available commercially. And these are primarily used in environmental sciences to look at different types of metals, but they actually do a really good job at measuring potassium and calcium in plants and in soils. And most of the work that's been done with this has been done using ground tissue that has been analyzed in cups right on the fruits, or right on the surface of this X-ray fluorescence window. But we've done work with whole apples to look at whether we can reliably measure potassium and calcium and how it links to bitter pit in an apple. And this is some of the work. So we compared the results we got from X-ray fluorescence on the y-axis with calcium measured with traditional ICP-MS on the x-axis, and we can see there's a really clear relationship between those two measurements. It means we're getting good resolution of what those calcium concentrations are actually in individual fruit. So each of these points is an individual fruit. And the X-ray fluorescence is an average of 4 spots around the fruit, and the calcium on the x-axis measured traditionally is the peel that is sampled right around the equator of the fruit. So you can see clear relationships with calcium, and we can also see clear relationships with potassium. And it's not just whole fruit, but we can actually then, because the measurement window is about 8 millimeters in diameter, we can actually map the surface potassium to calcium on the surface of the fruit, look at spatial variation on a whole fruit basis. And here we can see that, comparing bitter pit, a fruit affected by bitter pit compared to a healthy fruit, we see that a fruit affected by bitter pit has overall on average a higher potassium to calcium ratio, which we'd expect. But we also see a lot of variability within that individual fruit. And it's important to consider in terms of how you do your sampling protocols. One thing that the X-ray fluorescence, or the X-ray fluorometer, the portable one, allows us to do is to actually look at spatial variation within the tree. And because traditional analysis is difficult because you can't do as many replicates, there's a lot of processing involved in measuring those samples. So this experiment, we measured about 1,300 fruit across 9 orchards, and we mapped the distribution of those fruit within the tree. So we're able to assign a height in the canopy or a distance from the trunk, relative distance. So we see increasing in height or increasing in distance from the trunk. And overall, the higher you get in the tree, the higher or the lower the potassium to calcium ratio is. You can see that on the bottom here on the bottom right. And the potassium, you can see that potassium decreases as you go up the tree by a small amount, but calcium significantly increases. So we get about a 60 to 70% increase in calcium in fruit as you go to the top of the tree from the bottom of the tree. And this translates into differences in bitter pit incidence, where bitter pit is higher in the lower part of the tree for Honeycrisp than it is for the upper part of the tree. So by being able to increase our replication and interrogate spatial variation, we're able to answer different types of questions than we would be able to do with traditional analysis. There's also dealing with variability in the orchard. So I'm not going to cover this in depth, but one of the key factors, at least for our research on Honeycrisp, is crop load. So crop load can be highly variable, where you have trees that are on in their biennial cycle, and trees that are off in their biennial cycle, and then crop load of everywhere in between. And this really changes the bitter pit percent for those individual trees, and needs to be considered when you look at variability in the orchard. Some areas of the orchard might have really low crop load and have high bitter pit, and some might have some areas might have high crop load and low bitter pit. So how you sample and where you sample and how much you sample is really important in addressing that. And we'll get into that discussion, or get into that in the panel discussion. So I'm going to talk a little bit about measuring uptake and distribution in perennial fruit crops, and it's all about creating contrast. So we use stable or radioisotopes or analogs to create contrast, because I mentioned that there's so much pre-existing, the nutrient pool is so large, the pre-existing nutrient pool is so large that it's really difficult to see the small changes that happen in a 1 season or even a 2 season experiment. So we use these stable radioisotopes or analogs to create contrast. And for my examples, I'm going to demonstrate our use of 44 calcium, which is a stable isotope, and strontium or rubidium, which are analogs, to look at uptake. So we were curious about, well, timing of calcium spray applications, and really trying to measure how much of the calcium applied to the fruit surface via calcium sprays is absorbed when we apply it to the fruit. And to do that, because we see very, very small differences in bulk calcium changes when we do these experiments, we used a stable isotope applied to the fruit surface to look at how much of that isotope entered into the fruit at the end of the experiment. So when we measure the fruit, we get values of this delta 44 calcium. So it's really the amount of 44 calcium that has entered the fruit. And it's relative to a standard. So if the value is 0, it would tell us that there's no calcium that we applied in the form of this calcium chloride entering the fruit. At all 5 application points, the values are higher than 0. So we see both in the cortex and the peel. That's telling us that the calcium chloride is having an impact on fruit calcium. And it shows about a 25% total absorption efficiency on average. And only about 10% of the applied calcium enters into the fruit. And then only about, in a high density apple orchard, only about 10% of the calcium applied to the orchard actually reaches the fruit surface. So when you take 10% to 10%, that's 1% of the overall calcium that's applied to the orchard enters the fruit during that growing season. So very low efficiency in increasing calcium. So if you apply 15 kilograms per hectare over a course of a season of calcium, you'd expect 150 grams of calcium to end up in the fruit. And that really can't increase the fruit calcium concentrations by very much. It's going to improve them, but it's not going to solve the problem. And then looking at fruit surface temperatures, again, using the same methods, working on a USDA NIFA BARD with Dan Gamrosny and Haya Friedman, we can see that when fruit surface temperatures are low, calcium absorption is higher. So it's probably purely linked to drying time, where you decrease the drying time, or you increase the drying time, you increase the absorption of calcium into the fruit. We've done some analog tracing experiments using strontium rubidium compared to 15 nitrogen, which is kind of the standard for running these types of tracing experiments, where our recovery is high for both rubidium and 15 nitrogen, but strontium recovery is relatively low in the tree. So indicating that strontium is taken up at relatively low amounts. And this actually falls in line with some experiments we've done with 44 calcium as well. Looking at partitioning, we see that more strontium ends up in the roots than the shoots, compared to 15 nitrogen and rubidium, where rubidium, there's much more of the rubidium ending up in the above ground tissues, both the stem and the leaves. Indicating again that strontium is just slower in being taken up and moved through the tree. We also see some slight differences between cultivars and tracer recovery and distribution. We see that for Granny Smith, there's more 15 nitrogen ending up in the above ground tissue compared to the roots. And for Granny Smith, for strontium, we see that there's actually much more staying in the roots. So there's variability among cultivars in tracer recovery and distribution when run with this multi-tracer experiment. And lastly, I just want to quickly cover some work we've been doing on internal bitter pit and cellular structure. So this is with the microtomography X-ray CT line at Advanced Light Source. Here we have Fuji and Crisp Pink on the top and Honeycrisp on the bottom. You can where the light gray represents individual cells, and the dark gray areas represent airspace or porosity. And right away you can see Fuji and Crisp Pink, the fruit is more dense and the cells are much smaller. And Honeycrisp, the fruit is larger, so cell volume is almost 3 times higher for Honeycrisp than it is for these other varieties. And there's much more airspace as a result. So what gives Honeycrisp its characteristic crispness is its large, weak cells, which actually also probably contributes to its susceptibility to bitter pit, because those cells then become dilute in calcium, the cell walls, and they get stretched thin and they become weak. We can see a Honeycrisp with bitter pit having dead cell fragments scattered throughout the fruit here on the bottom right. And here you can see a larger bitter pit lesion. So we saw individual cells in the previous example. This is maybe 30 cells large. You can see this would be a bitter pit lesion forming just under the fruit surface. And then looking at whole fruit CT imaging, we can see that this sort of explains why the calyx end of the fruit has problems with calcium distribution, because most of the vasculature where calcium flows through is all on the stem end. So you see most of the vasculature is all on this upper half of the fruit and very little in the lower half. So it explains why there's lower amounts of calcium on the lower end of the fruit, because calcium, being relatively immobile in the plant, cannot move to be distributed to these lower locations. And we're looking at the relationship between bitter pit and density of this vasculature in Honeycrisp fruit. We also see that bitter pit is related to porosity. So more porous or bitter pit or Honeycrisp fruit that is affected by bitter pit is more porous. It has more airspace than Honeycrisp fruit that's healthy, of the exact same fruit size. So the more dense the fruit is and less airspace, the less susceptible it is to bitter pit. So just to kind of wrap it up, it's important to collaborate, reach outside traditional agronomy and horticulture, access these government user facilities. They're always looking for new applications for their tools, and they want to be as broad reaching as possible. They work with engineering groups, they work with medical research, and they also are working with a growing community of plant scientists. When doing nutrient research, it's important to create contrast in your experiments, and isotopes and analogs help distinguish and create contrast between newly acquired nutrients and the large pre-existing pools that exist in perennial fruit crops. And then lastly, minimize noise or account for that noise through your experimental design and mix models, where you account for random effects in your orchard that might add experimental noise or error noise to your experiments. So with that, I'd like to just thank my lab group, all the collaborators we've had, and the funding sources here outlined here, and then my wonderful lab group on the right. And I'd be happy to answer any questions. Thank you for your time. Thank you for listening, and I'm happy to discuss this further. Thank you.